good?
Yep.
Okay, great. All right. Okay. So now we're recording. Okay. Just that quick reminder to people that we will need to split up into different rooms. And so if your name is A through M here, N through Z at the other room, have ID of some kind with you. If you don't, we'll ask you to wait while we seat the students so we know should be in the right spot. If you arrive after 3:30, we'll ask you to wait till 3:40. And a couple of the students who may have time conflicts have written me. If you expect to be or you would like to be admitted after 3:40 and before 4 because of lab conflicts, I need an email to that effect. The proctors will have the names, and if the name is not on that list, you will not be admitted. We'll also ask you to let the proctors check your ID number against your answer sheet to keep down the numbers, so you'll make life easier for everybody if you have your ID matched up against your answer sheet to begin with, so that the matching up goes really quickly. Any questions about just the procedures of doing the exam on Monday? Heck of a way to do a Valentine weekend. Sorry about that. Okay. Let's head on to enzymes. And I just want to touch base on a very few of the slides that we looked at on Wednesday, sort of to reinforce the message of them. So, where are we? 7. So, slide 7 is making the point that in any reaction, you're going to have free energy associated with reactants and products. When there's a negative free energy, you can have a reaction go in that direction. That's why we call them the products. And getting between them, there's usually some kind of an energy hill, some transition energy that's associated with it. That's the barrier. That's the energetic barrier that keeps it from just happening instantaneously. And the amazing work of the enzyme is lowering the height of that transition free energy difference. That if the uncatalyzed reaction needs this much energy to get over that hill, an enzyme pulls this magic trick of finding a different way to get there, of going kind of part way up the hill and then down, rather than clear over the top. And then, if you look at slide 13, this is showing you that this principle of altering the free energy difference to the transition state is true even in covalent catalysis. So there are kinds of catalysis where you're just having non-covalent interactions going on, and that kind of a graph might be an easy representation for it. In other enzymes, there's actually points in which the substrate is covalently attached to the enzyme. And in that case, you're going to have a more complicated reaction coordinate because you start with reactants, you make some kind of covalent intermediate, a transition state to get to the covalent intermediate, and then a transition state to get to the products. But you still have the situation where the free energy change associated with that is less than the uncatalyzed reaction. So this principle of altering the free energy change of the transition along the way still applies in the case of covalent catalysis. And then on slide 21, 21 and 22 or 3 are going to look at sort of just refresh your memories as to what it is that enzymes do to make catalysis happen, to speed up that reaction, to lower that energy barrier. And one of the key things is that they can stabilize the transition state by comparison to that transition state free in solution. That is, if you look at the structure of the transition state, it's one that's awkward or strained or kind of funky looking, and that it would be unlikely or awkward to get into it in the absence of something intervening. And part of what the enzyme is doing is to stabilize and to provide a welcome environment for that transition state. And that's one of its key contributions. And this figure was just showing you how hydrogen bonds can play a key role in that. So you have hydrogen bonds to help to stabilize that tetrahedral intermediate. In fact, in this diagram overall, it'd be worthwhile to just take a moment to look through these different side chains in terms of how their chemistry contributes to all of this. That you have the carboxylic acid, you've got hydrogen bonding going on here, you've got an alcohol group on your serine making hydrogen bonds, and then the oxygen of that alcohol being actually the site of covalent attachment in a covalent intermediate. And then, so stabilizing the transition intermediate is one key thing. Some of the other kinds of things that enzymes can do, holding them nearby, which raises the effective concentration, holding them in a particular orientation so that the reactive parts are pointing at each other correctly, and that alone can account for a good 1,000-fold improvement over just the general environment. We talked about induced fit, and we're going to be seeing an example of that in just a second. We talked about electrostatic contributions, like tractor beams that draw the substrates in, and the fact that enzyme active sites can control the access to water. They may either permit one water to come in, as we saw in the case of chymotrypsin, or they may keep the water out completely and prevent the competing reactions, because water is everywhere, and water is going to do what it can do if it's given a chance. So let's look at another enzyme and its activities. And this is an enzyme called hexokinase. Hexokinase is going to reappear when we get into the metabolism of carbohydrates, and it will be one of the key steps in getting sugars into a form from which we can extract energy and so on. And this is an example of an induced fit. So here is hexokinase in its open configuration, and here is hexokinase after it has bound a glucose molecule. So you can see a very large change, almost like a clamshell closing onto that substrate. The active site in here has side chains. The side chains of the residues in those spots are going to be complementary to the binding of that substrate. And so even in the closed configuration, there are some polar groups inside because the substrate is a fairly polar substrate. And if you look at the structure of glucose, here is one of the ways to represent the structures of sugars. And you're going to see here that glucose is a polyalcohol. So here's an alcohol, there's another alcohol, there's another alcohol, there's another alcohol, there's an alcohol. So glucose is a very polar compound with 4 different alcohols. It interacts very strongly with water. And so one of the jobs of this clamshell closing on the glucose molecule is to strip off the water molecules that are like a shell around the glucose and would otherwise be the key thing that interacted with it. And by stripping away all that water, it also prevents some of the side reactions that would be otherwise likely to happen. The actual reaction that hexokinase performs is to bind an ATP and a glucose, and then to catalyze the transfer of one of the phosphate groups of adenosine triphosphate onto this carbon. So you've got glucose 6-phosphate. Here's the phosphate through a phosphate ester linkage. It's now charged, and you have an adenosine diphosphate, still a nucleotide, but just with 2 phosphate groups attached to it. We're going to see this reaction again. So I've been kind of speaking to this idea of structure reflects function, and the idea that the different biological molecules show signs of their evolutionary origins and so on. The proteases are a very powerful example of both of those principles. So this diagram here is based on X-ray structures. So they've crystallized the protein, shot X-rays through them. The atoms inside scatter the X-rays or don't. And you can then see the overall structure of the proteins. And what this is showing you is a spaghetti diagram for 3 proteases: chymotrypsin, which we've talked about already, trypsin, which you can hear kind of sounds like it, and elastase. And these are, in fact, a family of proteins that we can show are traceable back to a common ancestor. Their primary sequences have a lot of differences. The tertiary structure is very similar. And so this is rather like what we saw with myoglobin and hemoglobin, where the tertiary structure could be very similar, the primary structure quite different. And in this case, since this is an enzyme, what we see is the asp-his-seer, the catalytic triad, is completely conserved. And so those residues are exactly the same. Other residues are similar perhaps, but not identical. And then out on the surface, where the only job that the side chain has is interact with water, there's a lot of variation. Lots of polar, lots of charged things can mix around, mix and match, doesn't matter. Those surface residues that are just interacting with water vary the most. Now this asp-his-seer triad, this catalytic triad, performs its work, its catalysis, in that pocket where the substrate is bound. What's fascinating is finding out that if you look at other proteases, you can find that there are other proteases that use the same chemical strategy. So here's a different protease called subtilisin, because it's from B. subtilis, Bacillus subtilis. And if we were to go back, just sort of flick back and forth between those things, I think you can see those are quite different tertiary structures. So the tertiary structures are quite different. And as you might expect, there's no similarity at the level of the primary sequence either. However, the asp-his-seer here is the same catalytic triad. Same catalytic triad. So this is a case of parallel invention, or sometimes called convergent evolution. That those three side chains are just really good at doing that. And it was the solution to the problem in multiple cases. There are actually 5 families of serine proteases that have this asp-his-seer or DHS triad, but the packaging around it, the three-dimensional packaging around it, is quite different. 5 separate cases that converged on this solution. Any questions about that? Okay. Let's wait on specificity for a second, and if Eli and AJ would help me, I'm going to ask you to write some study questions. You chat with your neighbor about what kind of review questions you think would be helpful for preparing for the exam on Monday. I will compile those tonight and post those on Icon. If you'd like us to know that you were here today, feel free to print your name on the card, and we'll keep track of that too. If you do last name first in the print version, that's nice. It's a little easier to alphabetize then. But feel free if you want to discuss with each other possible study questions or write one off the top of your head. Question? No. I hand out the exam. It's really more valuable for you to work out the answers. And if you have, one of the things we might want to investigate is whether we could open a chat room, for instance, on Icon, and people could chat together about it. If you have specific questions where you're pretty sure of the answer, but you're not completely sure, certainly feel free to ask us. But in general, it's more valuable to work it out than to have the answer to. Okay. In case you're curious about how these cards are used, they don't give you points. There's no points attached to them. We keep track of them, and if someone is at the borderline at the end of the semester, it pushes you off the borderline. They don't generate numbers in the usual sense. No, you need more? Eli, do you need more cards?
No, he's counting. I'm just counting.
Okay. That's weird. Want to see something really weird? Okay.
Yeah, I know. I've noticed that before. I don't know why.
How come this machine can't?
You
Okay.
can't get that.
Yeah, well, I'll, for future reference, I guess. Cards. Oh, great. Does anybody still need a card that would like one? If you haven't got any questions you can think of and you'd still like us to have your name, you can just give us your name. You're not compelled to write a question, you're just invited. Who would like more time to write the question you're in the middle of? Okay, a little bit more time then. It's what? Okay. It's what? Video thing ate up a bit. Okay, if you could then finish writing your questions, that's great. If you've finished, please start sending your cards that way and forward. And if you still need another minute or two, take the time, and we'll make sure we get it before we leave. Let's go on thinking about enzymes and how they work as little molecular machines. So let's go on at slide 27. We've talked about catalysis, about accelerating the rate of a reaction by making the transition state more stable, by bringing things together, by pointing them, and so on. That's catalysis. That's how fast an enzyme goes. But the other amazing property of enzymes compared to inorganic catalysts is they are specific. And the specificity of enzymes is pretty amazing. Chymotrypsin is a moderately specific enzyme. Most of the enzymes we'll talk about during the rest of the semester are very specific enzymes. And their ability to enzymes to be more specific, orders of magnitude more specific than inorganic catalysts is an important property, just as their ability to be a catalyst is an important property. So let's take a little bit of a look at how specificity happens. And here is a comparison. You know, we just talked about how chymotrypsin and trypsin and elastase have similar tertiary structures. They have the same catalytic triad, and yet they do different things. That's specificity. Chymotrypsin tends to cut proteins after a large hydrophobic amino acid. Trypsin tends to cut after either lysine or arginine. And elastase tends to cut after small things, alanine, glycine, valine, stuff like that. So what is it about the structure of these things that allows them to have the same catalytic properties and different specificity. So now we're sort of piecing apart two different aspects of an enzyme. And this diagram, I think, does a nice job of comparing them. There's a pocket, a specificity pocket in these enzymes that determines what side chains will or won't fit well into that enzyme. So at the top of each of these structures is what they call the sessile bond. So this is the peptide bond that can be broken, can be cut like scissors. So the sessile bond is up here, up there, up there. So the sessile bond is up a little bit near that catalytic triad. But in binding the peptide, there's other stuff happening on the other side of the peptide backbone. There's a place for the side chain of the peptide to bind. And that specificity pocket has different structural properties in different enzymes, and therefore they are themselves have different specificities. So in chymotrypsin, which tends to cut after large hydrophobic amino acids, it's a fairly large pocket, and it's got inside of it 2 glycines and a serine. Serine is an alcohol. It's not terrifically hydrophilic. It's not like a charged group or something. It's tolerated by the large hydrophobic groups. Trypsin, by contrast, is the one that cuts after lysine or arginine. Lysine and arginine both have pretty large side chains, but they also have side chains with a full positive charge on them. And when we look at the bottom of the specificity pocket, we see an aspartate in there with a negative charge. And so that negative charge interacts to make an electrostatic interaction with the positive side chains of the arginine or lysine, and therefore the trypsin has a strong preference for that position in the peptide. And then if we look at elastase, which likes to cut after small hydrophobic things, the pocket is almost filled up with sort of hydrophobic or not very hydrophilic things. So instead of having the deep pocket of a chymotrypsin, it has a shallow pocket where the valine is sticking into it and the threonine is pointing. You can see it's pointing its hydrocarbon side. Threonine has, some hydrocarbon and some alcohol. It's pointing its alcohol away from the side chain. And so with a specificity pocket shaped like that, elastase is going to prefer the small hydrophobic side chains. So a very cute trick. Catalytic residues contribute to the rate of catalysis. Specificity pocket contributes to who is the target, who is the substrate. Now, chymotrypsin is not ready to go right off. Chymotrypsin actually has to be processed in order to take its full active form. And this is a strategy that the body uses in a number of different contexts of having a way to have something on hand in an inactive form and then processing it out where it's not going to hurt anything. Here's chymotrypsinogen. So if you look at it, up to there it looks like chymotrypsin, but it's chymotrypsinogen, and the ogen tells you this is something that can give rise to chymotrypsin. So this is a precursor for it. And chymotrypsinogen is not active. It will not have catalytic activity. It's got to be cut at least once before it starts to be active. And in sort of this kind of taking in each other's laundry approach, trypsin, the active enzyme trypsin, can cut at one of those spots to cleave the peptide chain there. This partially processed chymotrypsin is now enzymatically active. Perhaps not fully active, but it's starting to be active. So a molecule of chymotrypsin can cut another molecule of chymotrypsin to process out this little blue peptide there. So you now have that chunk out of it. And then 2 more cuts have to be made, removing the little green chunk, until you have the final alpha chymotrypsin, which is the fully processed, fully active enzyme. So it's an enzyme that actually has 3 peptide chains in it, but it came from 1 to begin with. Gosh, this is very strange. All right. So what kind of things are happening at the structural level during that processing? Well, this is a little interesting. Here is the precursor. This is the chymotrypsinogen, and these 2 little peptides here, there and there, are the short dipeptides that are cut out of chymotrypsinogen to make the fully functional enzyme. And if you look at the structure of the chymotrypsinogen, the tertiary structure is very close to the full mature protein. And if you look at it, you even see that the ASP/HIS/SER side chains are in the correct position. So here's your histidine ring and here's your serine alcohol. They're right where they should be to do catalysis. But until these little dipeptides are taken out, it doesn't have the proper specificity pocket. It doesn't have that oxyanion hole that stabilizes the transition state intermediate. So it's kind of like having a doorstop to keep the door from banging open in the wind or something. Now, you may think proteases, obviously they're interesting for digestion, otherwise we wouldn't get any protein out of our food. But proteases have a large number of other roles as well. It's really worth understanding even something as simple as a protease, and I'll just talk about one of those now, and that is the clotting cascade. The clotting cascade is something where you really want it to be on when it's on and off when it's off, or you would end up either bleeding to death or clotting to death. And both of those things can happen to people. And it turns out that it's proteases that control both turning on and turning off the process of clotting. Over here, what we have is a scanning electron micrograph that's been colored nicely to show you what an early stage of a clot looks like. And it's this mixture of protein fibers and little cell fragments called platelets. And you can see that a meshwork like that would be a good thing to kind of fill in a hole with. On the other hand, you don't want to fill in your capillaries, you don't want to fill in your lungs. So you have to have some way to make sure that this only happens where it should happen and doesn't happen elsewhere. And part of the trick is that the fibers here that you see are made of something called fibrin, but they come from a protein called fibrinogen. So the ogen, the thing giving rise to fibrin again. Fibrinogen is a soluble protein. You've got fibrinogen in your blood right now. A protease called thrombin, and that just literally means clotting enzyme. Thrombus, a thrombus is a clot. The protease thrombin can cut out little bits of the fibrinogen, and now you have a fibrin monomer, and those fibrin monomers can associate with each other to make an extensive bundle that then starts to build up the clot. The fibrin polymers are more like an intermediate filament than they are like a microtubule or a microfilament, because these things do not require ATP breakdown or anything like that. But the other thing that is important is that there is a cascade of enzymes that allows to have the amplification of a signal. And we're going to see cascades and amplification. Well, we saw it briefly already. We're going to see it in more detail when we come back to metabolism. So if our goal down here is to turn fibrinogen into fibrin, and active thrombin is what does that, if we had some way to control how many molecules of thrombin we have, that would be a way to control the size of the signal. And it's a multi-step proteolysis and activation process. So the enzyme thrombin, the active thrombin, can activate factor 9 to 9A for active, and that is itself a protease which can activate, sorry, that's 11. It activates 9 or factor 7. Those then are activated proteases. It can activate factor 10 or factor 7 and produce more active 10, and then that can activate more prothrombin into thrombin. So this is a case where a little bit of thrombin feeds back through this cascade to make a lot of thrombin. So it's a way to take a small signal and make it into a big one. Since these are enzymes, each of these are proteases and enzymes, each time, each of these arrows is saying, having made this protease, it can make dozens or hundreds of molecules at the next step. So you have a hundredfold amplification here and then another hundredfold. You can see you can get a very large single amplification out of a 3 or 4 step cascade. And so if you have something where a local event triggers the making of a little bit of thrombin, it can then have a local pool of cascading proteases that are going to make a lot more right there and not have it happening all over the body. There's a truly terrible way to die called disseminated intravascular coagulation, where the local control breaks down and all the thrombin in the body is activated. And basically, as you might guess, most of your body clots. And it's not a pretty way to go. Another general principle is that if there's a way to turn something on, there will be a way. You can see in your handouts that the font looks good there. So my secretary and I had no way to know. It's going to look like this on the screen. Another general principle in biology is if there's a way to turn it on, there's a way to turn it off. There are very few unregulated, go-forever processes in biology. And so, as we might expect, there are protease inhibitors. And there are some that have some very cute ways of doing that. Some of the naturally occurring protease inhibitors act by binding at the active site, interfering with the catalytic residues themselves. And in some cases, they do so with very high affinity. And this is an example here of trypsin interacting with bovine trypsin inhibitor, binding at the active site to prevent it from acting there. The importance of controlling both proteolysis and the inhibition of proteolysis is especially vivid when we look at elastase and emphysema. Elastase is one of those three members of the first protease family we talked about. Emphysema is a disease of the lungs, and it is possible to have emphysema just from the bad luck of the draw. More likely, if you have emphysema, you probably were a smoker. And part of what we're going to look at here is why that should be so. What kind of a chemical basis is there for that? And let's start by looking at what emphysema looks like. This is a nice, healthy, happy lung. You see these large spaces here that are nicely inflated, where you can breathe in deeply and inflate it and have your capillaries have full exchange, oxygen exchange with your hemoglobin and so on. And here is the kind of tatty-ratty, eaten-up looking lung tissue of someone who's suffering from emphysema. Clearly, this is not going to be something that's going to inflate well. It's going to be hard to get your oxygen fully into those hemoglobin molecules. It's going to be hard to get rid of carbon dioxide. In general, our bodies are under attack at a low-level way a lot of the time. So we can undergo damage and repair on an ongoing basis in lots of parts of our bodies. And in the body, elastase is one of the proteins that takes a repair role in the lungs. That is, it will go in, clean up a place where, maybe you had a really bad flu, right? And you've got some damaged spots in your lungs, and you need to clean out the gunk, that awful coughing up, getting rid of gunk stuff stage. Well, the elastase is helping to clean up some of that stuff. Under normal conditions, there's some alpha-1 antiprotease bound to that elastase to keep it under control, to keep it from going overboard. A few people are missing that protein genetically. And the people who are missing that protein genetically are at a much higher risk of having emphysema just from the bad luck of the draw. The other way that protease inhibitor can be modified, unfortunately, is by smoking. And this is showing you here how small a change is involved. This is the showing you the alpha-1 protease inhibitor. Here's the relevant methionine. And in the normal protein, the side chain methionine looks like this. If you're a smoker, it is in many cases been oxidized to a sulfoxide here. And if this damage to the protein has happened, it no longer inhibits elastase. And so the elastase goes overboard. It goes well beyond what's needed in the repair process and starts just creating more damage than it fixed. So both a testimony to the recuperative powers of our normal body and an example of the really multifold ways that smoking damages our bodies. I'd like to close up with something that's not actually in your textbook, but it is fairly recent findings. Let me again. I wish they, no way to check this. Okay. As we've gained more information about the structure of enzymes and the active sites and how they actually work, we've also gained insight into the evolution of enzymes and get insights that may help us in time to design new enzymes. And part of what that comes from is looking at the relative contributions of the catalytic residues, like the catalytic triad in the proteases, and the rest of the protein that plays a role in specificity and holding the whole thing together. And so if you compare two enzymes that are related across time, you can analyze the overall structure of that protein into what they're calling here a scaffold, just sort of the massive part that you need to hold everything together, you know, that bulky bit. Catalytic residues, so this would be like the asp-his-seer triad, for instance, in a protease. And then what they're calling variable loops. These are variable regions that can modify what those catalytic residues have access to. So these variable loops are going to modify what binds there, how it's oriented, and so on. And this kind of thing can show up at the level of the DNA itself. You can see regions of constancy, and you can see regions that are, so for instance, here they're showing you that the catalytic residues are preserved in both of those structures, whereas these variable loops that involve with specificity, not so much. And we already know that the genome has a certain amount of kind of fluidity about it. That is, it happens at a reasonable rate, different for different organisms. So you have copies of genes that are made just from the process of replication. And so once you have spare copies, you can have other things happening to those spare copies. So having on hand spare copies that you don't need for the original purpose, that scaffold there can have the variable loops assembled in different combinations. And as those different combinations are tried out, sometimes that will generate an enzyme with a new specificity. The overall catalysis has been preserved, but what it's doing it to will be a new substrate by recombining bits and pieces of the variable regions to get something new out of it that way. And this is giving us insight into the evolutionary origins of some of the enzymes we know well now. It has even been able to be tested to some extent in the lab. And this is, there are papers on this if you're interested in following up on it. So what they're comparing are two enzymes, one a human enzyme and one a bacterial enzymes, that have, generally speaking, the same kind of scaffold, bulk part of the overall structure. They have similar catalytic residues, and then they have quite different variable regions that are going to select different substrates. And so what they were able to do was to mimic this evolutionary process by making an undifferentiated scaffold by taking that part of the genome and allowing for variations to occur and putting in, for instance, substrates that would be available to the cell if it could come up with those branches and stuff, and off of that undifferentiated scaffold, recovering enzymes with different specificities in the organisms afterwards. And I think that's our last slide. Would people like to stay and have me answer some of the study questions, or is it 4:15 on a Friday? Make sure we get the cards, okay? Make sure you can leave the cards here. AV guy, we can save the file. Was my mic on? Okay. I was staring at it afterwards. So. feels so reluctant to do anything while he's doing something. I guess this is this him saving?
That's your sound.
Okay. Yes. Oh, okay. Okay.